I am Charles Cobbs, the director of the Ivy Center here for brain tumors, and it's a great pleasure to welcome Linda Liau, Dr. Liau from UCLA, to come join us. She is someone who I've admired for my career because she, like me, has, well, much better than I have, succeeded in combining a career in neurosurgery and basic and translational research that has led to an impact on patient care for glioblastoma primarily. Linda is the professor and interim chair of the Department of Neurosurgery at UCLA. She got her BS degree in biochemistry and a BA degree in political science at Brown. She then went to Stanford and got an M.D. in '91 and a Ph.D. in neuroscience in 1999 at UCLA. She completed her residency at UCLA and is now a tenured professor at UCLA. Among many of the things that she has focused on over the years, she's been very well known for being one of the early neurosurgeons and neuro-oncology Ph.D.s involved in immunotherapy. For those of you all who don't follow cancer and brain tumor research, perhaps as I do, cancer research has known about immunotherapy for decades, but in the last 5 or 10 years, there's been an explosion, basically exponential growth of interest in the field, and several clinical trials at our institution are going on now, and including one that was started by Dr. Liau. Her interest resided originally in taking patients' own white blood cells, isolating the dendritic cells, which are the antigen-presenting cells, and exposing them to antigens in hopes that they could be redirected, if given back to the patient, to target the tumor. And that led to a DC-Vax clinical trial that we've participated in. Lynn is also the editor-in-chief of a journal that I serve on the editorial board, Journal of Neuro-Oncology, and she trained, and her first postdoc was Rob Prinz, who's a PhD who's been here and given a talk last year. So it's with great pleasure I would like to introduce Dr. Linda Liau to give her talk. Thank you.
Thank you and good morning. It's a pleasure being here. This is the first time I've been here to SNI, and it's a beautiful facility. Kind of cold outside, though, I must say. So my talk is on immunotherapy for brain tumors, more specifically tumor vaccines. I just want to start with my disclosures. I do get research support from Northwest Biotherapeutics, although I am not a shareholder, and I also consult for Arbor Pharmaceuticals. So as far as just as an overview and a background for immunotherapy, there are kind of two main forms of immunotherapy, what we call active immunotherapy and passive immunotherapy. Passive immunotherapy, it has to do with, for instance, the adoptive transfer of T cells to the host, where you activate the T cells and then you infuse it back to the host. And these are, could be in the form of CAR Ts and other forms of activated T cells. That's not what I'm going to talk about today. What I'm going to talk about is active immunotherapy. And active immunotherapy actually requires the host to mount his or her own immune response. And there are some potential advantages to active immunotherapy, the most of which is some of which are that it can induce a de novo response to antigen. So you actually present an antigen to a patient and they mount their own immune response. It can induce systemic immunity. And importantly, it can induce dynamic immune recognition via a concept called epitope spreading. So for instance, if an immune cell kills a tumor cell, or targets an antigen on the tumor cell dies off. Other immune cells come in and pick up other antigens that are not necessarily the antigen that you initially targeted. But the holy grail for cancer and for cancer immunotherapy is really the last one, the ability to induce long-term immunologic memory. As you know, all of you know, glioblastomas and brain cancer are easy tumors, to treat initially. You could take them out, you could do radiation and chemo. The problem is that they keep coming back, and that's really the issue in terms of trying to induce some sort of long-term immunologic memory for it to prevent them from coming back. So over the years, there have been, hundreds and hundreds of preclinical studies and early phase 1, phase 2 clinical trials. But there have been, 3 tumor vaccine approaches to glioblastoma that have kind of advanced beyond phase 1, 2, and 2 multi-center phase 2 or phase 3 trials. One of which is the EGFR mutant peptide vaccine called Rindopepimut. And the issues with that is that it does require mutant EGFR status for the tumor, so that's only relevant in about 30 to 40% of glioblastomas. There was a phase 1 trial, and I'll talk a little bit about that trial, which was terminated earlier this year due to being unlikely to meet primary overall survival endpoints. But there are some issues in terms of trial design that I think we've learned some lessons from. There are peptide-loaded dendritic cells. So these are dendritic cells loaded with, 3, 4 candidate peptides. It does require pre-selection of the patients with appropriate HLA types because there are only certain antigens that have been identified for certain HLA types. There was a phase 2 trial that also did not meet its primary endpoint, although they did identify some subgroups that may have had some benefit. And then the trial that I've been involved in for a long time now is tumor lysate-loaded dendritic cell vaccines, or DCVax-L, which is what the company called it after we licensed it to them. And what that is it involves the patient's own tumor tissue and as the antigen source to pulse dendritic cells. And the advantages of that is that you don't actually need to know the antigen, because my personal feeling is that for glioblastomas, they're so heterogeneous that it's not going to be 1 or 2 or 3 or even 6 antigens. It's actually hundreds to thousands of antigens that we really need to identify to mount an effective immune response. We have been in a phase 1 clinical trial. It actually just closed enrollment last month, November. The enrollment included 331 patients, and we're just waiting for the number of events in order to do the data lock-in analysis. So hopefully we'll get that data soon. So just briefly on the EGFR V3 peptide vaccine, which may have been heard about or participated in. The phase 3 trials were really predicated on these earlier phase 1, phase 2 trials, which did show, let's see, don't know if there's a pointer. Well, it did show the, as you can see, the median survival versus of the treated patients versus the institutional controls. There was a difference between 15 months and 26 months, which is quite impressive. And this was work done by John Sampson at Duke. So that led to a phase 3 study of this EGFR V3 vaccine in newly diagnosed glioblastoma patients, which was called ACT4. This was an international randomized double-blinded placebo-controlled phase 3 study. Newly diagnosed surgically resected EGFR V3 positive glioblastomas, half got the vaccine, half got control with KLH and temozolomide. It was opened in 200 clinical trial sites across 22 countries, and there were 745 EGFR positive patients enrolled, so a fairly large trial. And the overall endpoint was overall survival. They completed enrollment in December 2014, and then in March of 2016, the interim analysis was conducted, and it was deemed by an independent DSMB that this trial was unlikely to meet its safety endpoints. So actually, the subsequent trials that were planned were stopped. And so why was that? What happened? And I think this is a lesson for us, and I'll talk about some lessons I've learned with our own trials as well. But one of the issues is obviously selection bias from early phase trials, because a lot of these trials, as much as you want to non-select, obviously you do have this propensity to select for patients who may or may do better, and those tend to be the patients that get on clinical trials. So when for instance, this phase 2 data, when you compared treated patients, open label treated patients to controls, you actually do see a significant difference if you do even historical matched controls. But then when and you could see that at the median survival, there is a significant difference between 2 years and almost 4 years in that cohort. But the issue sometimes is that median progression-free survival and overall survival may not capture the true efficacy of immunotherapy. This is actually a trial done in recurrent GBM, and you could see if you look at median survival, that's really the time at the 50% point when these two curves are analyzed, and you can see it's not really that much different. It's somewhere between 14 months and 15 months. So you could get non-statistical significance there. But I think the true benefit of immunotherapy is really out here. It's really the tail end of the curve. Because what a lot of us are seeing is that there are 20 to almost even 30, 40% of patients that do live significantly longer, meaning 3 years or more with immunotherapy. So the problem with the current design for FDA approval is that you submit a trial, the endpoints are difference in, for instance, overall survival or median survival. You see some difference, and if you're lucky, they're statistically significant, but you're not really capturing this tail end. But in reality, when you treat patients, if you give them the option, if you tell them you can live 3 more months with this therapy versus you have a 25 to 30% chance of living 5 more years, most patients actually would opt for that 30% chance of living 5 more years than okay, I'll do this and I'll give you 3 more months. But that's not really captured in our clinical trial design, because it's really designed to measure differences in chemotherapy and other therapeutics. And immune therapy it's a different field, and that's something I think we're struggling with now in the field to really get these drugs approved, given what we're seeing in terms of what the true efficacy for these agents are. So the lesson learned from this trial, I would say, is that yes, there is a selection bias for early phase trials, and we have to understand that. So when you power your subsequent phase trials, power them, to an extent where you could hopefully overcome that. But also there's a need for relevant endpoints. Median survival or overall survival at 1 year or even 2 years is really, may not be the meaningful endpoint for these trials. It's really that long-term tail end. The hazard ratio at 3 years or 4 years is really what we should be computing for these trials. And then also, I think as with any treatment, there's going to be a subset of patients that benefit and some that do not. Because obviously with these immunotherapy trials, 25, 30% have that long-term survival, but obviously the other 75% doesn't. So, I think the next goal is really to figure out who those 25% of patients are, either by, expression analysis or different, whether it's, extent of resection or, GBM subgroups. Those are the kind of things that I think will help move the field forward in glioblastoma. So with that in mind, I just wanted to give an update on the current status of our dendritic cell-based vaccine trial, which is called DC-Vax-L. just as an overview for those who are not familiar with what dendritic cells are, they are professional antigen-presenting cells. They're actually normal cells in your body. They come from the bone marrow. They're recruited to the peripheral organs, whether it be the skin, the gut, or anywhere where your body would capture antigen. And they reside there. They're in relatively small populations, but that's where they live. Once they capture an antigen, let's say a virus or a bacteria, then they're activated, and then they migrate to secondary lymphoid organs like the lymph nodes and the spleen, where they activate T cells, and it's the activated T cells that actually are targeting the antigen. And in our case, we want them to target tumor antigens. So this is the concept. The dendritic cell picks up tumor-targeted proteins. I guess the caveat in brain tumors is that these dendritic cells aren't circulating around in large numbers in the brain per se. So the thought of I guess the idea that we came up with in terms of this vaccine was that we take out the patient's own dendritic cells, take out the tumor, have them meet ex vivo, and then inject it back into patients in vivo. So we would take out dendritic cells, load them with tumor antigens, and there are various different types of antigens that you can load these tumors with. And believe me, we've tried many, many different types, and we still come back to the autologous tumor antigens just simply because we haven't found a good cocktail of antigens that really would work for this. And I think it's because of the heterogeneity of glioblastomas. So once they encounter a T cell, the T cell gets activated, as I mentioned. The activated T cells then proliferate and divide. There's this clonal expansion, and then they migrate to the tumor site, and they are able to migrate into the brain tumors. They are able to cross the blood-brain barrier into tumors, and we actually know that for a fact now. Interestingly, no one believed that about 20 years ago. So this was actually our first paper kind of showing that you could actually, and this was in a mouse model, that you could actually vaccinate against a target tumor antigen. And we did show T cell infiltration following vaccination into brain tumors. And this is actually very well known now, but back in the mid-90s, I remember applying for grants for this, and everyone thought, there's no way T cells get into the brain. It's immune privileged. So this is, I think, a change in the conception of how we think of immunotherapy. So actually in 1999, 98 actually, but we didn't get the paper out till 99. At 2000, we did the first human injection of DC-based vaccines into a human patient, and what that involved was taking out peripheral blood monocytes from the patient, culturing it with GM-CSF and IL-4, co-culturing it with tumor lysates taken from the patient. And over the years, we've tried different other tumor antigens to do this, and then injecting it back into patients. That first single case study led to a phase 1 clinical trial. And this is, essentially the concept that I was describing. We got some funding to do that phase 1 clinical trial, and what we found was that, yes, indeed, we are able, now in humans, we are able to induce a T cell response, T cells getting into the tumor following this vaccination. Interestingly, the T cell infiltration seemed to correlate with survival. So, for instance, the patient with the longer survival, the 30 months, had more T cell infiltration post-vaccination than the patients who actually did not have T cell infiltration. We also looked for other factors that correlated with response, and I could tell you we looked for many, many, many factors, and the only one actually that we found that was significant was actually TGF-beta. So the more TGF-beta you had in your tumor, the less likely you were getting T cell infiltration into the tumor, and the lower the survival. And that's probably because TGF-beta is acting in an immunosuppressive capacity there. So that went on. That led to a phase 1/2 clinical trial, and this was an open-label trial, and we saw good, just like other trials, a good extension of median survival compared to institutional historical controls. But again, this wasn't randomized. This was a phase 1 with historical controls. Although the 3-year survival percentage in our trial was 47%, and compared to our institutional controls, matched for age, extended resection, all that, which was only 21%. But as you can see, the difference is not necessarily, in retrospect, it's not the median survival, it's really that tail end. So that tail end in this particular case was almost 30%, and I could tell you a lot of those patients are actually still alive. So that's actually what intrigues me the most, who are these tail end patients, and how can we identify them? So with that, you get to thinking, well, what is different about these patients' tumors? So I'll tell you about 2 of the first, cohort of patients. So this was, patient number 1, and this was actually patient number 3 in the clinical trial. Both had, left temporal lobe tumors. Both underwent gross total resection. So, they had the minimal disease for prior to initiating this trial. They both had standard treatment with radiation and temozolomide, and then they subsequently enrolled in this clinical trial. And you could see the demographics are very similar between the two. The first patient was 39 years old, second patient 34, both male, good Karnofsky performance score. They both had small kids. When I met them, it was actually a very eerily similar type of patient. Histologically, if you looked at their pathology, they looked identical. You really couldn't tell anything on histology that was different between these 2 patients. But you could see the overall survival was significantly different. So the first patient, his overall survival was 33 months, almost 34 months, whereas the second patient was, at the time we published this paper, over 8 years. And actually, this paper was published quite a while ago. He's still alive now, going on 14 years. And we've had several of these patients. And this particular patient actually had activation of a CMV-directed T-cell response, and I'll show you that. But what was curious is, well, what is different about patient 1 versus patient 3? And one thing we did look at was gene expression profile. For those in the field, glioblastomas are now classified into different subgroups, proneural, classical, mesenchymal. So, one criticism of this trial is like, well, maybe you just enrolled all the proneural, patients, the good subgroup patients, and that's why you're getting longer survivals in these patients. So we looked at that, and interestingly, that wasn't actually the case. So patient number 1 was in that top cohort of patients. He lived 33 months, which was actually higher than the normal or average, median for glioblastoma, but it was actually not that much different than what would be expected for his subgroup, proneural subgroup, IDH1 mutated, MGMT methylated, those good prognostic factors. He kind of lived as long as probably would have been expected given his molecular subgroup. Interestingly, the patients who actually saw the most benefit from immunotherapy was in the mesenchymal subgroup, and that's actually the poorer subgroup. Those are the ones that are IDH1 wild type, MGMT not, methylated or non-methylated. It actually was in both populations. But those are the ones who typically have a median survival of about 14 to 16 months. So in that group, the vaccine actually did seem to show a significant difference, and actually all our long-term survivors were in the mesenchymal subgroup. So there's something about that particular subgroup that I think is inducing a better perhaps immune response for these patients, and that's kind of what we've been studying for the last 5 or 6 years now. And so things that seem to correlate with this is that they do seem to be increased with increased tumor-infiltrating lymphocytes. In our initial trial, we saw that those with increased lymphocytic infiltration do better. So it seems to be the case for this, but it wasn't actually just the lymphocytic infiltrates after vaccination, because we didn't, many of these patients actually didn't recur, so we actually have never gone back to take out tumor. But it's actually the lymphocytic infiltration before vaccination. So patients who tended to have some T cells in their tumor before entering the trials actually seemed to do better. So this is probably a more immune responsive subgroup, a subgroup where, there is some initial immune response and maybe it just wasn't enough, and immune therapy could make a difference in this particular subgroup. This also tends to be the group, and we're seeing this now in other trials, this tends to be the group that tends to show more, T2 changes following vaccination or immune enhancement following vaccination, and more likely, what we call pseudoprogression following treatment. So there is some, immune, component to this particular subgroup of patients. And interestingly, in more recent data, when we've actually done genetic sequencing of these tumors, these happen to be the most highly mutated. So interestingly, they're probably not, these patients are probably not doing as well because their tumors are so mutated, but they actually turn out to be a better target for immune therapies, because the more mutations you have, the more targets you have for immune-based therapies. So this is kind of the the quantification of the what we saw in terms of tumor-infiltrating lymphocytes. So it does seem that tumor-infiltrating lymphocyte content, pre-treatment, does seem to be a biomarker for survival post-treatment. CD8, yeah. CD4 there was some increase, but this is specifically CD8. So it's the T cell, the killer T cell infiltrates. And this is work done by Rob Prinz and who worked with me through all of this actually for the last 20 years, showing that because right now we're looking for biomarkers to figure out who that 25 to 30% of patients are. So this actually shows that an increase in TCR, T cell receptor overlap in the tumor versus the peripheral blood is also a potential biomarker for increased survival. So what this means, so what we did was we actually, in these patients, we isolated the T cells from the tumor, and we isolated T cells from the blood, and then sequenced the T cells and looked for their T cell receptor repertoires. So if we saw, so the patients that did the best were the patients who had the greatest overlap between the two. So basically, the blood T cell receptors were targeting the blood, and the tumor T cell receptors were overlapped, were similar. So the thought is that obviously you vaccinate to an antigen, and it's an antigen that's relevant in the tumor, because obviously why would you have T cell receptors to a particular antigen in the tumor unless there's something there that led the T cells to be there? And if your blood T cells had a greater overlap, that could predict a better immune response. And there were patients that were significantly, this is a confusing graph, but essentially what it means is the colors, the yellow is more overlap, and then the dark, hollow spaces meant there was less overlap.
So the left is the good.
Right. Right. The left is the good, and the right, and it's the colors. Ideally this, I should have made this into a 3D graph. It would have been easier in terms of the overlap. But what we're looking for is something that we can monitor in patients that allows us to monitor the response to treatment. So you can imagine if you take out the T cells, sequence the T cells, figure out the repertoire, T cell repertoire of the tumor infiltrating T cells, and then if you get a increase in that overlap following vaccination, you could actually monitor that, and if that increase goes down, then that's the time that maybe you need to revaccinate or boost that immune response to this T cell repertoire. Other things we're looking at right now is ways to enhance the T cell response, because obviously one issue is that the T cells may get in, but they're non-functional because of immune checkpoints and things like that. And what we found is that as or as you may have heard, things like these PD-1 inhibitors are actually now FDA approved for other cancers like melanoma and lung cancer. There have been trials in glioblastoma, but so far nothing substantial, I think, as a single agent. And I think the reason is that glioblastomas inherently aren't that immunogenic. You're not getting a huge population of T cells there. So if you don't have T cells in the tumor, then unblocking the block, because that's what these checkpoint inhibitors do, they actually block the immune suppression. Unblocking the block doesn't really help because there's no traffic going through. So that's kind of probably why these checkpoint inhibitors in itself does not work. So we actually have a clinical trial that we just got FDA approved for. We're probably starting right after the new year. To do DC vaccination followed by nivolumab, which is a PD-1 inhibitor. And this is just based on some preclinical studies in animal models showing that if you combine the two, we do get increased survival, and probably for that reason that even though the vaccine can get T cells in, it's still the T cells are immunosuppressed by the local immune environment. So going back to, so we've also looked at these long-term survivors. What is it about them that makes them such long-term survivors? And this was actually patient number 3 in our phase 1 cohort. And what we did was and he happened to be HLA-A2 positive. So we have, antigens candidate antigens that we kind of tested his T cells against. So we just, tested for a panel of known candidate antigens. And interestingly, for a lot of these patients, we actually used viral antigens as the control to test for, whether there was a immune response. And as it turns out, this patient actually mounted a very significant immune response following vaccination to CMV. And then when we went back and looked at his tumor tissue, he actually had very strong expression of CMV on immunohistochemistry. And then if you look at the graphs before treatment, these are tetramer analysis of T cell population. So before treatment, he had 19%, very small percentage of his T cells were recognizing CMV. After treatment, that went up significantly to 4.36%. And that's huge, to have 4.6% of your T cells throughout your body targeted to one particular antigen. So there was a huge increase in the immune response to CMV following vaccination. And then it waned over time. At 6 months, it was low. And then when we started doing booster vaccinations, and as we do boosters, it goes up again, wanes in 6 months and goes up. And then we kept doing that until we actually ran out of vaccine for this patient. But this paper was published in 2008, and he was a 5-year survivor at that time. He's still alive today, and it's been 6 years since. So he's actually it's 9 years since that time. So he's 14 years out now, and his scan, I can tell you, looks exactly like this. He actually has no recurrence, and when I first met him, his kid was his son was very small. Now his son's in high school, and hoping to go to his high school graduation soon. So this is obviously the issue that we want to learn more about. And over the years, there have been many clinical trials using dendritic cell vaccines throughout the world. And this was a review done by Rick Comator back in 2013. And at that time, there were already 21 published studies, 403 patients in 5 continents. And overall, the conclusion of this meta-analysis was that the dendritic cells loaded with autologous tumor cells did increase overall survival in glioblastoma patients, pretty much in all these trials. They did show some survival advantage. Granted, these were all non-randomized single-arm studies, so that's, you have to take these results with that in mind. So based on all the earlier trials and the preclinical work, we went on to embark on a phase 3 multi-center randomized clinical trial, which actually just closed enrollment last month. And what that involved was, we do surgery on these patients. So patients underwent surgery. Following surgery, they got a leukapheresis to isolate their white blood cells. We would culture them with GM-CSF and IL-4 to make them dendritic cells. And during that time, they would undergo standard treatment, which is radiation and Temodar. It takes a few weeks to make the cells, and then after that, the patients would have their post-radiation MRI scan. And then they'd randomize to either placebo or injection on these in this clinical trial. so there are a couple things that, we designed this trial about almost 8 years ago now. So when there are a couple things that I think we've learned from this trial that may affect the final results, and I think it has helped us to kind of just learn more about how to design future trials in the future. One issue was that at this post radiation MRI scan, if the patients had progressive tumor, they were actually excluded from the trial. They were put on into this separate informational arm. And the thought about that was that, in our prior experience with the phase 1 trials is that if you had big bulky disease, or actually not even bulky disease, progressive disease, those patients did not respond as well, because if your tumor is growing, exponentially, you don't have enough time to mount an immune response to the tumor, and those patients, at least what we saw, did not benefit. So that was the rationale for excluding that, the what we call the early progressors in that trial. Unfortunately, what we've learned over the years is that determination of early progression is difficult because of the issue of pseudoprogression. So there may be some patients that were excluded that probably should not have been excluded, or vice versa. So the trial was held in 50 sites throughout the United States, Canada, UK, and Germany. It was actually well over 50 million at this point. And as you can see, your site here, that's that little star up in Washington, way up north. And we just recently closed the enrollment. So what this was an international randomized double-blinded placebo-controlled study. The randomization was 2-3, randomly assigned to DC-Vax versus placebo-controlled, and a third just got autologous PBMCs, no dendritic cells, but they did get an injection of their own blood. The primary endpoint was progression-free survival, overall survival of secondary endpoint. We've had 331 subjects enrolled, and currently the trial is still blinded until data lock, and data lock will occur when 75% of the patients have an event, meaning death or progression or death. So far that hasn't happened, but so the whole group is doing better. The treatment and the control group, they seem to be doing better than the historical, analysis for this. In a way, it's good. It's very good for the patients, but it may not be so good for the trial because you want to see a difference between the two groups, and the fact that the whole group seems to be doing better, although we don't know who's who, that could be an issue in terms of FDA approval. But we all do believe that there is, efficacy in this, and it's really comes down to what potentially happened with the EGFR vaccine trial, in terms of the endpoints and the trial design and how to best elucidate the differences here. So this was the trial design. So patients underwent surgery, and then, during that recovery week period post-surgery, they had their leukapheresis. Then they had the 6 weeks of radiation and Temodar while the vaccine was being made, and then they had their baseline MRI. So at that baseline MRI, if they had progression, they were put into something called the informational arm. They still got the vaccine because we made it for them, and it would be unfair to not give it to patients, but they weren't included in the trial analysis. So the trial was randomized, and that's probably why it took a little longer to accrue, because we had some attrition at this point. The trial was randomized 2-1. And that was the intent to treat population. They either got DC-Vax or placebo. And then this is the issue with why we think perhaps the whole group is doing better. There is a crossover arm. So if the patients got progression or was deemed to have progression on the MRI scan, they were allowed to cross over and get the true vaccine if they were on placebo. The patients love this. It's much easier to enroll patients with the crossover arm than to say, if you're randomized to placebo, too bad. But it really does kind of muddy the trial results, because then essentially we may be just comparing DC-Vax early versus DC-Vax late. So here's here's the treatment groups. Again, we're still blinded to the results, but what we do know is 2/3 were randomized to DC-Vax and a third to placebo. So all the boxes in green got DC-Vax at some point. So obviously the top part of this chart, those were the patients who randomized to the treatment. And at this point in looking at their analysis, they either had no progression, they either had progression and recurrence and went on some other treatment, or they had progression and recurrence and then just went on standard treatment and continued with their vaccine. The placebo arm, there was a percentage of those patients, like I said, because of the crossover arm, that got progression, that got the DC-Vax at recurrence. And it turns out that the green boxes accounted for 86% of the patients. So a significant percent of these patients actually did get the DC-Vax at some point. And I think it's, so we went back and we looked at this informational arm. So these are the rapid progressors. You know, this was these people that got taken out early because their tumors were growing despite radiation and chemo. And they got enrolled into the informational arm or the pseudo-progression arm. You know, we kind of called it slightly different things during these two time points between 2008 and 2012, and then subsequently we felt that some of these were pseudoprogressors. But looking at this arm, there were 55 patients, so this is almost like a separate little single arm study. What we found was that 20 had further progression on subsequent MRI. So 20 were probably true progressors. So they were the ones that they had the progression on the MRI scan, and the next scan showed more progression and more progression. So those are true progressors. 25 had stable disease at the 2-month scan. So you got that first post-radiation scan, and then you had another scan and actually was stable. So those, in retrospect, were probably pseudoprogressors or people that, you know, were read by the radiologist as progression, but were pseudoprogressors. And the issue with these multi-center trials is that they're all centrally reviewed in terms of the radiology review. That's to, you know, keep it unbiased. But because of the central radiology review, you don't have the benefit of the clinical picture to decide whether this is true progression or pseudoprogression. So it's all based on, you know, a certain percentage of increased mass on the MRI scan. So I think what's happened is, you know, there is a group of patients here that are pseudoprogressors, and then one patient was a confirmed pseudoprogressor because his tumor actually totally went away. So we knew, you know, he was definitely a pseudoprogressor. And then 5 patients we couldn't really tell for sure with the subsequent MRI scans. So of these patients, because they were off trial, we were able to analyze their data early. And this is essentially what that data shows. Of the 25 patients that were pseudoprogressors or possibly pseudoprogressors, 10 of them are still alive over 3 years. They've progressed. They haven't had any, not even alive. They actually, you know, most of these haven't had progression. So it's actually quite exciting to us that, you know, we hopefully can identify a population of patients where this could have a significant benefit, meaning 3-year or more survival for this patient. So that's that tail end of the curve, and probably has to do with, you know, the patients that do have more pseudoprogression may actually do better. We'll obviously do all these subgroup analyses once the trial, the bigger trial, is unblinded and we have the cohorts identified. There's you know, countless different subgroups that we were looking at, and you know, one of which is actually looking at CMV to see if now that we have 331 patients, we have a population of those patients that are going to be long-term survivors. To see what is it about, you know, their tumors or their immune response. We have blood on all these patients through all these time points. So that would be a very interesting analysis, I think. So the survival of this informational arm, these 55 patients, as you can see, the rapid progressors, so those were the 20 patients that had that initial read as progression, so they got taken off the trial. The subsequent scans showed further progression, so they were kind of confirmed as rapid progressors. Those we kind of knew weren't going to do as well. But the ones that were pseudoprogressors actually did significantly better, and they're drawing out that tail end of the curve. 25% of them are actually now over 4 years. So that's I think some intriguing preliminary data, and once we get the bigger randomized trial unblinded, we obviously like to look at that further. So in conclusion, brain tumor vaccines are feasible and safe and can potentially lead to a significant subgroup of long-term, meaning greater than 5-year survivor, progression-free survivors. So this is just not being alive. This is being alive without progression. And I think, for patients that is meaningful. Granted, it's not everybody. Like I said, it's about 25%. But if we could identify who those 25% of patients are to get on these trials, that may be meaningful. The lessons learned over the years from our trial and the other trials that have been going on in brain tumor immunotherapy and vaccines is that there is definitely a selection bias with early phased non-randomized trials. So we have to account for that. The endpoints that are relevant for immunotherapy are probably going to be different than the kind of accepted endpoints currently that are being established for FDA approval of chemotherapies and other agents. And then the heterogeneity of the tumor and the patient population needs to be taken into account. So as I mentioned, we at least in our early trials, we see some difference between the mesenchymal subgroup and the proneural subgroup of glioblastoma. That's obviously something we're going to look at in our randomized trials when that gets unblinded. Things like MGMT methylation, which tends to portend to more pseudoprogression. That's obviously something that we would look at, and that's something very easy to do. It's done. Everyone looks at the MGMT methylation now, so that's actually a biomarker that potentially could be already universally applied. IDH1 mutation, IP19Q code deletion, TERT mutations, these are other things that would play into the subgroup analysis. And then Ben Ellingson, who works at UCLA with us, he's been doing a lot of work on image analysis and looking for imaging biomarkers for this and other types of treatments. So I just want to say thank you for your time. And last night at dinner, we got to talking about, how much tax I pay in Southern California. 13.9% state income tax. So I make 13.9% less than all of you every year. But the reason I think we pay for that tax is that this is my son and my daughter. We could go surfing and skiing on the same day. So. And it doesn't rain much. Actually, we need rain. We're in a drought, so I wish we did rain. So thank you.